AI-designed protein boosts T cells to kill cancer. Imagine a world where AI-designed proteins can help our immune system defeat cancer. Sounds like science fiction, right? But here's where it gets groundbreaking. A recent study has turned this vision into reality. Cancer immunotherapies heavily depend on T cells, which activate by interacting with the major histocompatibility complex, MHC, on cell surfaces. MHC presents fragments of intracellular proteins, including cancer antigens. Scientists have long sought to enhance this interaction but lacked an efficient method. Enter Timothy Jenkins, a data scientist, and Sine Hadrup, an immunologist, both from the Technical University of Denmark. They leveraged AI to create novel T-cell receptor, TCR-like proteins that specifically bind to predetermined cancer antigens based on their known crystal structures in complex with MHC. In a groundbreaking study published in Science, they introduced a designer protein termed minibinder, which significantly aided T-cells in killing cancer cells in vitro. Remarkably, this discovery took mere weeks, a stark contrast to the typical 18-month timeline. But this is the part most people miss. The researchers also demonstrated that their platform could function even for targets with unknown crystal structures. "This is a remarkably cool paper," exclaimed Christopher Klebanoff, an oncologist and immunobiologist at Memorial Sloan Kettering Cancer Center, who was not part of the study. An incredibly innovative use of generative AI to tackle a deeply challenging issue in the field. To validate their tool, the team designed a protein targeting NY-ESO1, a well-known cancer-associated antigen. Hadrup explained their choice, stating, "We selected NY-ESO1 because its crystal structure with MHC is already known, ensuring the target's structure wouldn't hinder the computational process." The platform generated several minibinders, each uniquely different from typical T-cell receptors. The AI's output was astonishing. These minibinders bear no resemblance to standard T-cell receptors. Klebanoff noted, the next challenge was to confirm if these diverse minibinders could effectively mimic T-cell receptor functions. Using AlphaFold2, the team predicted the MHC binding traits of 44 minibinders and validated their binding and specificity in vitro. One minibinder stood out, showing the strongest binding to MHC. Cryo-electron microscopy confirmed a high correlation between the computational models and the actual crystal structure. Moreover, T cells engineered to express this minibinder significantly outperformed unmodified T cells in killing NY-ESO1 positive melanoma cells. What's incredibly promising is the strong correlation between our lab results and computational predictions, Hadrup emphasized. Pushing the boundaries further, the team tackled a more complex task, designing minibinders for a metastatic melanoma neoantigen with an unsolved MHC-bound crystal structure. They successfully developed proteins that bound strongly to the neoantigen in vitro. Despite initial skepticism around de novo protein design, the 2024 Nobel Prize in Chemistry has highlighted its immense potential. This is just the tip of the iceberg for de novo protein design applications. Jenkins stated, "The ultimate goal is to see this work in clinical settings, not just as an academic curiosity." Jenkins added, discussing future plans. Hadrup also hinted at broader applications, such as developing these molecules as soluble binders to block MHC and T-cell interactions in autoimmune diseases. These therapeutic areas remain unexplored, and we aim to demonstrate their potential in the future. She concluded, "So what do you think? Is AI the future of cancer treatment, or are there unforeseen risks? Share your thoughts." UW researchers discover AI-powered breakthrough for precision cancer treatment. Imagine a cure for cancer that's as precise as a GPS tracker, homing in on rogue cells while sparing healthy ones. Researchers at the University of Washington just cracked the code, and it's about to change the game. Here's why this matters to you. Current treatments can be like firing a shotgun when you need a sniper rifle, damaging innocent targets. Now, AI has enabled a breakthrough that could create biological foglands, guiding immune cells directly to diseased ones. Breakthrough alert. Nobel laureate David Baker's team, yes, that David Baker, who solves protein puzzles for Nobel Prizes, just unveiled an AI-driven method to design proteins that act as neon signs for immune cells. These custom-built biomolecules highlight cancerous or virus-infected cells, helping T cells, think biological FBI agents, find and destroy the enemy. The science is published in today's issue of Science, and the implications? Massive. Here's the kicker. This isn't just theoretical. Working with labs at Stanford University and Memorial Sloan Kettering Cancer Center, the team used artificial intelligence tools called RF diffusion and protein MPNN to engineer proteins targeting specific cell surface markers. Picture this. Each cell has unique ID tags called peptides on its surface. Healthy cells display normal tags. Cancer cells, or virus-infected ones, show mutations or viral fragments. By designing proteins that recognize these abnormal tags, researchers can create homing devices for immune cells. So how did they test this? The scientists designed proteins for 11 different targets, including dangerous strands from HIV and cancer-related molecules. Out of these, 8 showed they could effectively guide immune cells to attack. 2 of these designs triggered such a strong response that T cells obliterated the target cells. Think of it as programming immune cells to recognize and eliminate specific threats. But what's next? Bingshu Liu, a lead researcher, says the team plans to spin this into a real-world treatment through a new company. The Institute for Protein Design has already spawned 10 startups since 2014, with Baker co-founding 21 companies. This could make personalized medicine cheaper and faster. As graduate student Julia Bonzanini notes, I'm hopeful this will lead to therapies accessible to patients worldwide who aren't helped by current treatments. One major advantage? Flexibility. The team modified successful proteins within days using AI to target new cancer mutations and viruses. Need a protein to recognize a rare cancer variant? Adapt it rapidly. This adaptability could lower costs and democratize access to customized treatments. But here's where it gets controversial. Will these therapies only benefit the privileged? How can we ensure equitable access? Debate it in comments. Do AI-driven personalized treatments risk widening the chasm between different socioeconomic groups? Bonus info for the deep divers. The open source software they developed is freely available online, allowing researchers globally to build upon their work. Other authors hail from UW's multiple departments, Stanford's Garcia Lab, and NSKCC's Scheinberg Lab. A true cross-institutional collaboration. So what's the takeaway? We're witnessing the dawn of a new era where AI doesn't just predict disease, but actively redesigns the tools to fight it. As Baker puts it, our immune systems work incredibly hard, but sometimes they miss the subtle clues cancer leaves behind. These computer-designed proteins act as signposts, making those clues impossible to ignore. But we're still in early days. How do you feel about AI playing such a central role in healthcare? Could this breakthrough signal the beginning of a new chapter in medicine or introduce unforeseen consequences? Sound off below. Revolutionary AI platform designs molecular missiles to attack cancer cells. Imagine a world where precision cancer treatment isn't just a distant dream, but a tangible reality, thanks to groundbreaking advancements in artificial intelligence. Researchers have unveiled an AI platform capable of engineering protein components to supercharge the body's immune cells, turning them into molecular missiles that seek out and destroy cancer cells. This innovative approach, detailed in a recent study published in Science, marks a monumental leap forward in cancer immunotherapy. Traditionally, developing treatments that harness the power of the immune system has been a painstakingly slow process, often taking years to identify suitable molecules. However, this new method slashes that timeline to mere weeks. As Associate Professor Timothy P. Jenkins from the Technical University of Denmark explains, we're essentially giving the immune system a pair of new glasses, allowing it to spot cancer cells more accurately and efficiently than ever before. The AI platform, a collaborative effort between DTU and the American Scripps Research Institute, addresses a significant hurdle in cancer immunotherapy: the difficulty in targeting tumor cells without harming healthy tissue. Typically, T cells recognize cancer cells by binding to specific protein fragments or peptides displayed on the cell surface via PMHC molecules. However, this natural process is fraught with variability and complexity, making it challenging to create personalized treatments. To overcome these obstacles, The researchers employed advanced computational techniques to design custom protein binders that can lock onto cancer-specific PMHC molecules. In their study, they successfully engineered a mini-binder that tightly binds to NY-ESO1, a protein found in various types of cancer. When this protein was introduced into T cells, it transformed them into impact T cells, unique immune cell products capable of selectively attacking cancer cells in laboratory settings. It was truly exhilarating to witness these computer-generated mini-binders perform so effectively in real-world conditions, shares postdoc Christopher Hauren Johansen, co-author of the study. But here's where it gets even more exciting. The team didn't stop there. They applied their AI pipeline to design binders for a cancer target identified in a metastatic melanoma patient, demonstrating the versatility and adaptability of their method. This breakthrough suggests that the platform could be instrumental in developing personalized immunotherapies for previously unknown cancer markers. One of the standout features of this research is the incorporation of a virtual safety check. Using AI, the team screened their designed mini-binders to ensure they wouldn't inadvertently react with PMHC molecules on healthy cells, thereby minimizing the risk of harmful side effects. Precision in cancer treatment is paramount, emphasizes DTU professor Sean Recker-Hadrup. By addressing potential cross-reactions during the design phase, we've significantly increased the chances of creating both safe and effective therapies. While this technology represents a significant step forward, it's important to note that translating these findings into clinical applications will require further refinement and testing. According to Jenkins, it could be up to 5 years before the new method is ready for human clinical trials. Once available, the treatment process would involve extracting immune cells from a patient's blood, modifying them from the lab to carry the AI-designed mini-binders, and then reintroducing them into the body to target and eliminate cancer cells. And this is the part most people miss. The potential implications of this research extend far beyond cancer treatment. By harnessing the power of AI to engineer bespoke molecular solutions, we open up new possibilities for tackling other complex diseases. But what do you think? Could AI-driven precision medicine revolutionize healthcare as we know it? Share your thoughts in the comments below.